Welcome to the fascinating world of insect vectors. Imagine you are a city planner trying to stop a network of rogue delivery trucks from distributing harmful packages across your city. To stop them effectively, you first need to understand exactly what kinds of trucks are being used, how fast they drive, and how they are built. In the biological world, these delivery trucks are specific groups of insects, and the harmful packages are disease-causing pathogens. Today, we will explore how their unique features drive disease transmission. Now that we understand our goal, let us look at how we categorize these biological delivery trucks. In biological taxonomy, an order is a major classification rank just below class. It groups organisms that share a highly similar evolutionary and anatomical blueprint. Why does this matter? Because by understanding the unique anatomical and physiological features of these vector orders, we can figure out exactly how to dismantle their disease transmission networks. If we do not know how an insect is built or how it behaves, we cannot effectively control it or protect vulnerable populations. Building on that classification, medical entomology primarily focuses on four major groups of insect vectors. These are Diptera, which includes true flies and mosquitoes, Siphonoptera, which are the fleas, Siphunculata, known as sucking lice, and Hemiptera, the true bugs. Each of these orders has evolved highly specialized anatomical and physiological features to feed on blood. Inadvertently, this blood-feeding behavior is exactly what allows them to transmit devastating diseases. Let us dive deeper into the specific physical traits that make each of these orders such effective vectors. Let us examine the first two orders closely. Dipterans, like mosquitoes, are incredibly mobile because they possess one pair of functional wings. They also have highly adapted piercing sucking or sponging mouthparts designed for rapid feeding. On the other hand, siphonapterans, or fleas, take a completely different evolutionary approach. They are wingless and laterally flattened, which allows them to glide smoothly through the fur of their hosts. Instead of flying, they have developed powerful hind legs for jumping onto hosts from the ground or from animal nests. Moving on to the next two orders, we see even more specialized adaptations. Siphunculata, the sucking lice, are also wingless like fleas, but they are dorso-ventrally flattened, meaning they are flat from top to bottom. They are equipped with specialized claws that allow them to grip host hair tightly, making them excellent permanent residents on a host. Hemipterans, the true bugs, are built differently. They have partially hardened wings and a robust, segmented beak that is specifically designed for piercing deep into tissue to extract blood. Now that we have discussed their features, let us look at them visually. In the top left, you can see the mosquito, representing Diptera, highlighting its single pair of wings and long, piercing proboscis. In the top right, the flea represents Siphonoptera. Notice its laterally flattened body and those enlarged hind legs built for jumping. On the bottom left, the louse represents Siphunculata, showing its dorso-ventrally flattened shape and specialized gripping claws. Finally, on the bottom right, the kissing bug represents Hemiptera, displaying its partially hardened wings and segmented pucing beak. These physical differences dictate how they interact with hosts. Seeing these physical differences brings us to a crucial point. Why does this anatomy matter for disease control? Understanding these specific anatomical and physiological features is absolutely critical for public health. For example, knowing that wingless fleas live in the nests of rodents allows us to target our chemical treatments directly at rodent burrows, rather than uselessly spraying the open air. Similarly, understanding that flying mosquitoes require stagnant water for their larval stage allows us to use cultural control methods, like draining puddles, to break their life cycle. This biological knowledge leads us to a powerful mathematical tool. Scientists use a concept called vectorial capacity to measure how effectively a specific insect order can spread a disease based on its biological traits. The formula is C equals M times A squared times P to the power of N, all divided by the negative natural logarithm of P. Here, C is the average number of new infections generated by one infected host per day. The variable M stands for vector density. The symbol A represents the biting rate, which is squared because the insect must bite twice to transmit disease. The variable P is the daily survival rate, and N is the incubation period. Notice that if the biting rate increases, the overall capacity increases dramatically because it is squared. Let us apply this formula to a practical example involving a malaria-carrying mosquito. We are given a mosquito density of 10 per person, a high biting rate of 0.5 bites per day, a daily survival rate of 0.8, and an incubation period of 12 days. First, we square the biting rate, giving us 0.25. Next, we calculate the probability the mosquito survives the 12-day incubation period, which is 0.8 to the power of 12, or about 6.87%. Then, we find the daily mortality rate by taking the negative natural logarithm of 0.8, yielding 0.223. Plugging these into our formula, we find the final answer is approximately 0.77. This means every infected person generates about 0.77 new infectious mosquito bites per day. Now, let us compare the mosquito to a flea transmitting the plague. Because of a rodent infestation, the flea density is high at 20 per person. However, their biting rate on humans is lower, at 0.1 bites per day. Their tough bodies give a high survival rate of 0.9, and the bacteria incubates in just 5 days. Squaring the biting rate gives a very low 0.01, but because the incubation is short and survival is high, 0.9 to the power of 5 shows a massive 59% of fleas survive to become infectious. The mortality rate is the negative natural log of 0.9, which is 0.105. Calculating the final capacity, we get 1.12. This means one infected host leads to more than one new infection per day, indicating a rapidly growing outbreak. Having calculated both examples, let us reflect on what the math is telling us about their biology. Even though the flea's biting rate on humans was much lower than the mosquitoes, the flea still produced a higher vectorial capacity. Why? Because their tough, laterally flattened bodies give them an excellent survival rate, and their specific environment allowed for a very high population density. The mosquito relies on its flight and efficient piercing mouth parts to achieve a high biting rate. This perfectly illustrates how different evolutionary traits, whether it is wings for mobility or tough bodies for survival, directly impact the variables in our mathematical models, making both insects highly effective vectors under the right conditions. We have covered a lot of ground today, so let us review the key takeaways. First, we learned that insect orders like Diptera, Siphonaptera, Siphunculata, and Hemiptera possess highly specialized anatomical and physiological features that perfectly equip them to be efficient disease vectors. Second, we saw that these evolutionary traits, like wings, jumping legs, or flattened bodies, directly impact their biting rates and lifespans. Finally, we discovered that by translating these biological features into mathematical models, like vectorial capacity, we can accurately predict transmission risks and design targeted strategies to prevent devastating disease outbreaks. You now have a solid understanding of how insect anatomy drives global disease dynamics.